Hello, my name is Taylor Berg, and I am representing the Chase Lab at Casper College. Our research was focused on identifying uh, community-associated antibiotic-resistant genes, and we did this using the nanopore sequencing platform. And we started with a 2019 INBRE SPREM grant, and we had a few different questions we were trying to answer. Can we use the nanopore MinION to find ARGs or antibiotic-resistant genes? And are there reservoirs of genes in the environment? Our actual project started in the fall of 2019, but our experiment started in January of 2020. And that moves us on to what is nanopore MinION sequencing. And so this is an array of voltage pores that are arranged on a flow cell, and each of these pores are capable of sequencing a DNA reaction or an RNA reaction. And there are up to 2,000 pores that can be sequencing at the same time. With our project, we had usually around 1,200 that were sequencing at the same time, and we were able to sequence up to 400 base pairs per second. This slide shows the procedures that we did throughout this project. So the first one that we started with was the Kirby-Bauer method, and this was to find antibiotic resistance in our samples to begin with. And so we used different antibiotic discs, and as you can see in some of these pictures are not from our actual samples. These are just representations, but you can see the zones of inhibition. Sorry about that. You can see the zones of inhibition around the discs, and this shows which ones would be resistant or not. And so we started out with that to see what kind of antibiotics were resistant to our samples. And then we moved on to our second step, which was harvesting the metagenomic DNA from our samples. And we used this DNeasy Power Soil kit. And once we had our metagenomic DNA harvested from that, we used the NanoDrop spec to quantitate our data and see the quality of the DNA that we had harvested. And so we were looking for quality ratios of OD 260 to 230 and OD, sorry about that, OD 260 to 80. And then we moved on to our third step, which was actually sequencing and analyzing the DNA. And this is where the nanopore MinION device comes into play. And so we had to use 400 nanograms of our DNA that we had harvested, and we had to put it through a rapid sequencing reaction, which took about 30 minutes. And then we were able to load that DNA onto our flow cell, and we had that run for 48 hours. And after that, we were able to analyze the data from the nanopore MinION device, and it was uploaded to this platform down here on the bottom. And this next slide is going to show the results that we got from the nanopore MinION device. Oh goodness. So altogether, these are our overall results, and we were able to sequence about 6 gigabase pairs in the 5 samples that we tested, and the total sequences that were read per sample were between 150,000 to 450,000 reads, and then we also had an average read length over in this bottom right corner, and this just shows how long each length of the DNA was, and it was the average was between 2 to 20, as you can see down there. And then this is our results from the actual nanopore system, and we altogether were able to identify 300 plus CAR genes And out of all of those, 70 plus of them were 90% or above in accuracy. And then these two tables down here shows, the one on the left side is showing the just a sample of the total organisms that we were able to identify through this platform. And then the bottom right is showing just some of the different resistant genes that we were able to identify with that platform and system. And that is our results. And so in the end, we did prove that the MinION sequencer can work in identifying antibiotic resistant genes and reservoirs around the community and in the environment. And we can use this information. It could be used in a clinical setting. All right, and I would just like to say a quick thank you to the INBRE program, Dr. Chase, Casper College, and the co-authors on this project for working with me and throughout the duration of the experiment, and yeah, just making the research great.